Guys, so today, I'm gonna go over these osmosis notes with you again in case you need to see it. So you can pause the video right here if you want to, um, if you haven't copied these notes in your notebook yet. Remember, copying down your notes and entering them into Schoology under the notes assignments is part of a overall collective notes grade at the end of the 9 weeks. If you already copied this or you're ready to move forward, we'll go ahead and move forward. So what is osmosis? It's the diffusion of water across a semi-permeable membrane. We know that this is a semi-permeable membrane because you see this artificial membrane here. It's only allowing these particles to cross and not these bulkier particles to cross. So it's selective about what permeates through. It selects water, which is what this is, a water molecule, and not the bulky sugar molecules. And water, as you can see, moves from a high concentration of water to a low concentration of water until equilibrium is reached, right? So we have unequal concentrations across the membrane. Water is going to move where there's a lower concentration of water. Even though it looks even here, because there's more solute, because there's more little dots here, there's less room for water. And because there's less dots over here, there's more space for water. So water is still moving from high to low. But another way that we see this is for some students, it's easier to see that water moves to where there's more solute. So wherever there's more of these dots, that's not water. So osmosis, water moving across a semi-permeable membrane, moves to where there's more stuff, which example could be sugar. Anything that dissolves in water, sugar, salt. So it's moving to that end until water is equal on both sides. Even though it looks like this is completely unequal, the water on both sides is what's equal. So water will move until equilibrium for water is reached. So remember our discussion about solutions. A solute is what dissolves in the solvent, and our universal solvent is water. So Kool-Aid dissolving in water, solute dissolving in solvent, is a solution. Our cells live in solutions, whether they're in solutions in our bodies or in a body of water. So solutions affect cells. Water will move by osmosis. But the question is, which way is water going to move, inside or outside of the cell? That's going to depend on the environment and solution that you place that cell in. So there's three comparative words that we need to know: hypotonic, hypertonic, and isotonic. We use these words for comparison. What do I mean? Well, first let's look at this image here. We see that there's less solute here, right? Less dots. And on the other side, there's more dots or circles, whatever you want to call them, stuff that dissolves in water. Let's call it sugar. So less sugar, more sugar. So if I asked you to compare these two, you'd probably, in a description, you'd probably say something like, there's less sugar molecules on the left side compared to the right side. On the right side, there's more sugar molecules compared to the left side, right? Simple. What I'm saying now is that instead of using less and more in that simple description, we need to know how to use the words hypotonic and hypertonic. So we would say the left side is hypotonic compared to the right side. The right side is hypertonic compared to the hypotonic left side. So just replacing those less and more words. And we're talking about solute concentration here, okay? So isotonic is when comparing two solutions and they're equal on both sides. So we have three different environments that we place this cell in. In environment A, this cell is getting really big. In environment B, the cell looks like it's staying the same. And in environment C, the cell starts to shrivel up like a raisin. So we're going to use the words we just learned, hyper, hypo, and isotonic, to describe the environment that the cells are placed in. So we're comparing the environment to the inside of the cell. So a cell in a hypotonic environment, in a hypotonic solution, will swell up like a hippo, which is how we remember this. Hypotonic rhymes with hippo, hippo. So hypotonic solution. If the cell's getting big and big and big like a hippo, that's because the cell has more solute. Okay, the outside of the cell has less solute. So one way students remember where water moves is remembering this phrase, salt sucks. Salt sucks up water. So if there's more solute, for example, salt, inside of the cell, it's going to suck up all the water. So in a hypotonic solution, where is there more solute? Well, since it's getting big like a hippo, there's more salt in the cell because that's where all the water is getting sucked up to. So water enters the cell and leaves the solution it is in. So the cell swells hypo-hippo. In an isotonic solution, we can see water molecules moving in and out of the cell simultaneously at the same rate. So the solution and the cell have the same concentration. Water enters and leaves the cell at the same rate. So some way you can remember isotonic is balance and they're moving in and out is that this solution here is perfect for this cell here, for this animal red blood cell. So you can remember I'm so perfect for isotonic because water moves in as it moves out. So cell stays the same size. A cell in a hypertonic solution, you see water moving outside of the cell and it's getting all shrivelly like a raisin. So the cell has less solute because remember, salt sucks. It's all moving out of the cell. So where is there more salt? Outside the cell, which leaves the cell with less compared to the outside. Water leaves the cell and enters the solution. Okay. So note that the terms hypertonic, hypotonic, and isotonic are relative. They can be used only to express the relationship between a given solution and another solution separated by a membrane. So when you're asked to solve this for questions on osmosis in a test question or assignment question, it's always important to follow these steps. So first, make sure that you have inside and outside the cell that your solute and solvent add up to 100. So 90, 10, 100, 63, 37, 100, and then draw an arrow where you think water will go. Remember, salt sucks. So you look at that 37%, you know that's where water is going to get sucked up to. And since the water is moving out of the cell, now you can tell what kind of environment is the cell placed in. Well, since it's shriveling up like a raisin, it's probably hypertonic. So let's practice. Remembering that salt sucks. If you look at 10% solute versus 15% solute, the 15% solute is probably going to soak up all the water compared to the lesser amount 10, right? So it's going to move outside of the cell. Here, let's try looking at water instead of the solute because you can look at it both ways. We know that salt sucks, right? 10 versus 60. So that 60 is going to suck up all the water. So back to the practice questions. Here we have in the second beaker, we already said that this one was the where the water would move outside of the cell because salt sucks. And in here, if we're going off of the salt sucks again, where is there more salt? In other words, solute inside the cell. So that's where water is going to get sucked up to. But we need to be able to flip the switch and look at it from a different perspective. We know that water gets moved to where there's more solute, where there's more salt, right? An example of a solute or sugar. But water still moves by diffusion. It moves from high to low concentration. So if we were just to look at the water concentration, right? That's solute. But if we were to look at 40% compared to 90% water, which one is the higher amount of water? 90%, right? So that's the higher concentration, which would move to the low concentration because water naturally moves down or with the concentration gradient. So from high to low, you would still get the same answer if you were to use the other method, which is to remember that salt sucks and there's more salt inside the cell. So this third one, again, the same way you can look at the solute or you can look at the water. Let's start by looking at the water. We have 80% water outside versus 75% water inside the cell. So the higher concentration obviously is at 80. 80 is higher than 75. So from high to low, the water will move into the cell. And if you were to just flip the switch and look at the solute, the salt, right, it would still move inside the cell because 25 is greater than 20, and solute sucks up all the water. Okay, so solutes can show you where the water will go to, not by just looking at the concentration of water. Remember, hypotonic and hypertonic are comparative when comparing two solutions. And how exactly does a polar water molecule cross a non-polar portion of the membrane? Remember those hydrophobic tails in the middle of the membrane? Well, since it's not very friendly with water, water travels in bulk through an aquaporin, which is a channel protein. So osmosis is actually a type of facilitated diffusion. And then if you remember what we learned about turgor pressure, it's the force behind the cell walls of plants, which allows them to keep their shape. So the force comes from the water filled into the vacuole of the plant cell. And the vacuole fills up with water and presses up against the cell wall. This provides turgor pressure, which causes the plant to be structured and happy and healthy. So with that in mind, what type of environment do plant cells prefer? Do they prefer hypotonic or hypertonic? Well, plant cells like to get filled up with water. See, they want their vacuoles to be filled with water to provide turgor pressure and structure to the plant and the cell. So they prefer hypotonic. Remember, hypo rhymes with hippo. They like to get big like a hippo. However, with animal cells, this is not the case. Here we have the hypotonic environment providing the structure to this healthy looking plant, but the animal cell, on the other hand, it bursts. And when an animal cell bursts, this is called cytolysis. The reason an animal cell will burst when it fills up with water and not a plant cell is because animal cells don't have cell walls. We don't have cell walls, and plant cells do, so that protects them from bursting. In an isotonic solution, plant cells, it's not the preferred state for them to be in. It's not their preferred environment. For animal cells, this is our preferred environment. Water moves in and out simultaneously. The animal cell looks fine. The plant cell looks not as good as over here. And then with plasmolysis, what that means is the cell shrinks up and shrivels, and the plant doesn't like this, and animal cell doesn't like that condition. So that is it for the notes. Let me know if you need any help on anything at all.